So I have to say, I've been asked to do many talks in my time. I've never, ever been given a 40-minute slot to talk about all clinical trials in MS. And yeah, and it's one of these things I was asked, and I said, clinical trials in MS, and I kind of put it away to do after Christmas, and I kind of went back to work on the 2nd of January and thought, I have 2 weeks to prepare a talk on all clinical trials in MS. So it's a bit of a big undertaking, and then like it wasn't enough, they then said, could you do biomarkers while you're standing up there as well? And I've sat through 1-hour talks on biomarkers, and I've sat through 3 days of ECTRIMS where they describe ongoing trials in MS. So you're going to get all of that in 40 minutes. So a bit of a task. Right, disclosures. I have to say we're all a bit conflicted. I make these two look like virgins by the look of it. So yeah, sorry about that. But I am going to, probably worth putting up there, I'm going to talk about a lot of drugs made by a lot of companies. I don't think that my involvement with them has influenced the choice of this, and I hope you come away with the same impression. So what can we achieve? This is going to be a very high-level flyover MS research, okay? You're all coming at this from different levels. Some of you will be very familiar with some of these studies and very familiar with what is out there, and you might get quite a lot from the detail. For some of you, I have the feeling this is going to be a little bit, you know, like those photographs of someone's face that are made up of lots of little photographs of their face, and if you stand back long enough, you see one thing. Some of you may just see a bit of a big picture, okay, and that's fine. Usually I judge success in the talk if everyone stands up and applauds at the end. For this talk, I have to say, just most of you not falling asleep will do me fine. And I say in the middle, we're going to have a brief detour into neurofilaments. So I do worry about, I have a lot of slides. I've never tried to show 100 slides in 40 minutes before. We are going to run through it. It may sound a little like the shipping forecast, okay? For those of you, anyone watch Black Books and Tasman Grieg? Yeah, okay, so the shipping forecast isn't always bad, and only one person is laughing, okay? None of you look this up, okay? Right, so what did I have to do for this? I started with a blank sheet of paper and wrote down the studies I knew about, and that didn't feel very scientific. So what I've done is I've gone to clinicaltrials.gov. Now, if you don't know about this, if you're going to do a trial in human subjects, you should register that trial, and it only makes sense. If I were a drug company, I would do 8 trials in a drug. If 2 of them worked, I would publish those 2, and I would never tell anyone I did 6 negative trials. So what you should do if you're going to do a trial is tell everyone you're doing the trial, and that should be publicly searchable. So that's what clinicaltrials.gov is. It's run by the US Library of Medicine. You can go on and you can see any trial that's been set up in humans, and you can search it. So I searched for MS. I looked for studies that were active and not recruiting, were recruiting or not yet recruiting. So hopefully that ongoing or just around the corner area. Okay, and you come up with hundreds of hits, and just for you, I did go through them all, okay? Now, I'm not going to present them all, so I've skipped over some dull ones, okay? Pregnancy registers, extension studies, and everything which, in my opinion, was small, speculative, or dull, okay? So everything here has interested me, or I think might interest you, and you can determine if we've done that. So, what order are we going to do this? For a bit of structure, I'm going to go through the stages of MS and highlight some of the current trials which are going through, and then at the end we have bits on diet and we have bits on certain treatment strategies, and we'll see whether we can run through where we are by in the 35 minutes left to me. Radiologically isolated syndrome. Now, due to Gordon dropping out of the next step due to his minor illness that we're not allowed to talk about, diarrhea and vomiting, we're actually going to do a bit more on RIS in the next section. But suffice to say that this has been a topic we've never really known what to do when someone has come with lesions that look like MS, but they haven't declared themselves as having an illness. You will get this all the time. We're just talking before, I think as nurses, you're probably spared this a little bit because they come to us when they have headaches, and then we're asked, should I go on treatment? The answer is at the minute we don't know, but there are 2 big trials going on. One on teriflunomide, one in Tecfidera. You'll see similar design, about 200 in each of them, and they're both looking to see what is the first neurological event or progression. Those studies are both running, a couple of years to go, okay? So worth there. I have to say, if you're a Newcastle MS nurse and none of you are here, you do get to see these patients because you have these people that have had headaches. You say, I'm not going to be worried about you until you have your first attack. And then they say, what does an attack sound like? And I say, I have 4 people who, if you phone in your symptoms, will tell you whether what you have sounds like an attack or not, and will put your mind at rest if you're worried about MS. So I must admit, some of our RIS are actually with the MS nurses. So CIS, again, we're going to talk about this in the next bit. This is a rapidly vanishing area because with the new criteria, very few people now have CIS. But there's one trial out there that did interest me, which is happening in this. And this, we're going to talk about this with other ones. And this is a cell-based therapy. So you've heard a lot about Epstein-Barr yesterday from Gavin. And we know that Epstein-Barr lives in B cells. So what if you could kill those Epstein-Barr infected B cells? Now, there's various ways to do that. One is to get your own body to do it. Okay, so what you have, you've got CD8 cells, which are killer T cells. I take those out of you. I train them, I make them live with Epstein-Barr B cells long enough till they learn to hate each other, and then I put the T cells back into your body. They go and find your Epstein-Barr infected B cells and kill them. So it's a neat strategy, okay, and it's in trial with CIS. Okay, so that's a study that's being done. It's being done in Nantes in France. It's proof of concept only at the minute. The primary outcome will just be safety, but that's running in my report as early as next year, and that's kind of fun. So, the brief detour into pediatrics. By the way, I'll just say now, there's something called the Noun Project. Okay, I'm aware with the Belfast accent, I'll now spell that. That's N-O-U-N. Okay, the Noun Project, which is running, and if you want an icon on anything you want, you can find it. So, pediatrics, so we have a couple of studies going. Terry Kids, okay, it's been running for a little bit and is still running. This is teriflunomide versus placebo in children. We can discuss a little bit about the design of this. Obviously, we have fingolimod already with paradigms, with the results that have shown that went against Avonex. Teriflu is going against placebo, so it'll be interesting to see how those results are received. BG12, dimethyl fumarate, is in trial against Avonex. Again, similar group. You'll see kind of looking at kids over 10, under 18. Numbers are similar, both still running in a couple of years to go. And then of interest as well, so Lemtrada is being looked at in children. Okay, but to get into the Lemtrada kids study, you have to have failed the first-line therapy. You'll see it's a little bit small, but in a couple of years' time, 3-4 years, we're going to have a rough idea. We're going to have fingolimod with the trial, we'll have Teriflu, we'll have BG12, and we'll have Lemtrada. So a much clearer idea about what to do with children with MS. This is one which comes up in a couple of sections, and this is a different way of trying to manage MS. So again, as Trevor was saying, we know T cells have a big role in MS. Obviously, B cells do as well, but it seems to be a little bit that you can take either element out or you can take both out, and either might work. This strategy's been around for about, 10, 15 years now, which is that we know that the T cells that try to attack myelin, they've got unique receptors that recognize myelin, but those receptors themselves can be recognized, okay, because they're quite unique. So what if you designed something that would make cells attack cells that had receptors that were receptive for myelin? Okay, and that's what this group are doing. So they've taken T cell receptors, which recognize myelin. They vaccinate you with those so that you then develop an immune response against T cell receptors that recognize myelin, and then you kill your own T cells that recognize myelin. Okay, so this was being done. It ran for a while. The company that first started it went bankrupt. Okay, but this company has been taken over by Immune Response Biopharma, and the US FDA have given it orphan drug status for children and for secondary progressive MS. So this is one that I think everyone had thought had gone away and is maybe coming back again. Estimated to complete this year. I think it's held up, and I don't think it's running this fast. The primary outcomes are MR and white cell things, and they're doing this in kids, which is sort of interesting. Okay, right. Relapsing remitting disease. What new agents do we have? Okay. Now, this is where I've had to educate myself. Like everyone, pharmacology for me stopped when I left medical school, and then you kind of had to catch up afterwards. So there's these new groups of drugs now called the NIBs and the MIBs. Okay. The NIBs, okay, and In case I forget it, I've actually put the entire text up that I want to say on the slide. So NIB is short for inhibitor, okay? And a TNIB is a drug which inhibits a tyrosine kinase. So you've heard about tyrosine kinases already. These are molecules which live inside cells. Something binds to the outside, and to get the signal from the outside to the nucleus involves a signaling pathway. That signaling pathway often involves tyrosine kinases, and we can inhibit them. The tyrosine kinases are often specific to particular pathways and particular cells, okay? Now, the hematologists and oncologists have been using these for years because you can go in and kill certain cell types, okay? We're starting to use this now in MS. So the first NIB that we have is a TNIB, so it's going for tyrosine kinase, and it's Evolut tinib, okay? It's been made by Merck, and they have a phase 2 trial. I've kind of cheated a little bit. It's still on the clinical trials as active, although it's actually been completed and reported. So this drug was used against dimethyl fumarate. Very exciting positive outcome as a new small molecule that might work in relapsing remitting MS. They're moving that into a phase 3 trial, okay? But that one's kind of round the corner. That's the NIBs. The MIBs, okay, again, and a zomib is something that inhibits a protease. I think it's because it's a proteasome, okay, and I think that's where the zomib comes from, maybe, okay. So this is a zomib, again, being used in relapsing remitting disease. This is a London trial. I think it's Gavin's group. And what they're doing now, they've found one of the zomibs that currently is being used in myeloma. So myeloma is, you get this overgrowth of plasma cells that make antibodies. This one will go in and kill antibody-producing cells. Now, it's very interesting. If you look at the way ocrelizumab works, we often say that one of its strengths is that it doesn't kill plasma cells, okay, because plasma cells are the ones that are producing some of your long-term immunity. They're also the ones that produce oligoclonal bands. Okay, so what the London group are trying to see is, can we kill the plasma cells making the oligoclonal bands? And in fact, the primary outcome is how many people turn oligoclonal band negative. Okay, and they're looking at it across the sorts of MS, but they're going in to see, can we kill these cells? And if we do, what happens? So that's the next one round the corner. Okay, and then the weird and wacky, this is a Bosnian pharma company, okay, and they're the only people in the world looking at this particular product. It's called EK12. Your pituitary produces pro-opiomelanocortical hormone, which gets broken down to ACTH. ACTH makes your adrenals produce steroids. Steroids are good in MS. Okay, along that pathway, there are two peptides that get cut off the first molecule. One is metenkephalin, okay, which is an enkephalin, and one is tridecactide. They've taken these two peptides, they've put them into a magic combination, and they give you it 3 times a week subcutaneously, and they're trying it against rebif in 400 people. So this is no small undertaking, okay? So it could be that Bosnia leads the world. Okay, right, that's the small molecules, that's the new ones. What about relapses then, okay? Anything new coming for relapse? And the answer is yes. Mad people in Sweden, okay, are again looking at one of the NIBs, okay? It's a TNIB, and you already know a TNIB is going to be a molecule that inhibits a tyrosine kinase. This particular one seems to promote apoptosis in, I think, myeloid and lymphoid lines.
Shuts the blood-brain barrier and switches the peripheral immune to an anti-inflammatory phenotype.
There, you heard it first, okay? And the idea is here that we're going against methylprednisolone as an alternative way of treating relapse. Primary outcome will be functional score at 28 days, secondary outcome. So that's kind of exciting, a new way, maybe a better way of treating relapse. Okay, and here I'm going to mention ACTH gel for a while. So ACTH, I am old enough to remember patients that got ACTH for MS. Okay, it's Synacthin, it costs pence. Okay, and it was the first randomized trial that was done in MS, showed that ACTH was better than placebo for relapse. Okay, it was made by a company called Quester, now got about Mallinckrodot. Have you ever heard a name that sounds like an evil company? That would be it, okay? And they've patented this into a gel, okay? And they're looking at people that have failed on methylprednisolone. What if you gave them this magic gel a couple of weeks later, the primary outcome being EDSS? And I'm sorry, because this isn't a pharma-sponsored talk, I can now show you a little bit of this, okay? I'm not going to, not going to comment on the name of the blog, okay? But again, for those of you that know ACTH, it was pence. When this company bought it, they put the price up 1,000 times overnight. Okay, so what was something that was $40 is now $40,000. So you're going to see ACTH gel appear quite a lot for being tried, because let's face it, if you had a company that cost 40,000 per pot, you'd be trying it and everything, and they are. Okay, right. The lookalikes, okay? So we've talked about new agents, we've talked about relapses, and what are people doing that are kind of lookalikes. So we all know about ocrelizumab, but we've been impressed by the results. We're starting to use it, so it's not going to be surprising that other drugs aiming the same thing are coming along. So Asclepios 1 and 2, this is Novartis, ocrelizumab, okay? If you know your monoclonal antibodies, the earliest ones were the oximabs, like rituximab, which are chimeric. We then have ocrelizumab, which is in the middle, and then with the imumabs are fully humanized, okay? So this is a fully humanized anti-CD20, okay, offered to Mumab, okay, and it's being used in a study that looks very like the ocrelizumab study, and we're due to finish quite soon, actually, okay, with results coming out, but might give us an alternative way of getting rid of B cells. Right, ublituximab, okay, so this is like rituximab, so again, a chimeric, a slightly older style monoclonal antibody, again, getting B cells, but you'll see running in a very similar, slightly smaller trial to see. So we may have 3 anti-CD20s aimed at MS in the next couple of years, okay. Right, again, the lookalikes, fingolimod we've had for a long time. There are other IMODs out there. Ponesimod is an interesting one. It's been around for a long time. The phase 2 was very exciting, and particularly because this one doesn't seem to have some of the cardiac side effects that fingolimod had. So they were kind of running with it for a bit. It went on hold while the company moved around. It's now come in a very oddly designed study added into Tecfidera. So instead of going against Tecfidera, we're looking for people that are failing in DMF and then adding in ponesimod. You can only imagine how successful it is in terms of funding and safety monitoring to have a patient in both those drugs with the risk of lymphopenia, but they're still running with it, and that's out there. I haven't bought shares. Okay, there are, however, other IMODs out there. Ozanimod, okay, this was a company by Receptos, if you've been running around ectrems for a while, now bought out by Celgene. This has positive phase 3 trials already. It is ready for market. They made a mistake in their FDA submission and their refiling this year. There was some missing data, okay, and the open label extension is going there, but you may have another IMOD knocking on your door in the next couple of years. Of the interesting lookalikes, this is one which comes through. So as you know, if you've been using dimethyl fumarate, Biogen has spent a lot of time convincing us that this is not monomethyl fumarate, that it's not Fumiderm, that their safety profile is unique, that this is a complete molecule, that the only thing that's going to work and be safe in MF is dimethyl fumarate as Tecfidera. They're now working with a company producing direxol fumarate, which is metabolized to monomethyl fumarate inside, okay, and they are working on this together. What's interesting is, and I'm afraid I have no insight into this commercially, that there are only 2 trials listed. One is looking to see whether the GI events in the first month are the same, okay, to try and see if it's safer and better tolerated. And the other is an open label single group assignment to see how well it works, suggesting that if you, the only way I can put this together is they're looking to piggyback on the dimethyl fumarate data, say this is the same and doesn't cause the same GI. So you may hear Biogen trying to tell you that all monomethyl fumarates are a bit the same. I'm not sure. I'm sorry if that's anti-commercial and I get shot afterwards. Right, okay, relapsing remitting, okay, that's the kind of lookalikes, the news that come out, head-to-heads of current agents that are out. This one is very interesting, okay, kind of coming comparisons, rituximab versus dimethyl fumarate, okay? So this is potentially, it is running, it's a big study. It will give an answer as to whether this is as good or better, and would be the interesting one of rituximab as an unlicensed product, generic anti-CD20 coming in, and what will we going to do with the result of this when a drug which is now generically available has shown itself to be as good or better than another drug in a randomized controlled trial. So watch this space, that's kind of interesting. Okay, fingolimod versus dimethyl fumarate, okay, never really been done as a randomized controlled trial. We've got real world data, but the Italians are trying this to head, and that's kind of interesting, okay? And that's the head-to-heads. Happy? Okay, right, I'm still awake. Yeah, I'll have a little bit of a check that everyone's still there. Stem cell transplants, okay, now again, I'm probably going to undersell this. You had a talk yesterday on it, so I haven't gone on. The one registered trial I could find then is coming. It's a multi-center Scandinavian one looking at stem cell transplant against alemtuzumab, okay? Now, we've already had, I think you got the MIST trial presented yesterday. We know that STAR MS is coming through, but this one is still up and running, a low-intensity, non-ablative regimen. And what can I say? We all must be really excited about this when we've seen the early results. This will get a chance to see whether these are replicated in a different population. So, cell-based therapies in relapsing remitting MS. We've had the small molecules, we've had the monoclonals. What else is out there? So, this is an interesting one. So, I talked about with the kids about taking your CD8 cells out, training them to hate Epstein-Barr cells, and then giving them back to you. This is an off-the-peg one, okay? So if I take your cells, it's autologous. This is allogenic. So what they have is a kind of like O-negative group of cells, okay, that already hate Epstein-Barr cells. And if you have a rough genetic match with their panel, they will actually put these foreign cells into you. Now, the excitement of that is that taking it out from you, culturing them up and putting them back in again is long, it's expensive, there's risk of infection, there's all sorts of things can go wrong in that. Okay, this would be that literally we would have infusion bags of cell lines that will kill most people's Epstein-Barr B cells if you have a rough genetic match. So that's kind of interesting, okay? But the dendritic cell ones is kind of the next big one of cell-based therapies. Now, dendritic cells are close to my heart because I did my MD in dendritic cells, so it's great to see them coming around. Okay, my other big topic was B cells. Who predicted all of MS? Okay, anyway, so dendritic cells are cells that you have that kind of live around your body that are your sentinels, that they're kind of your spies that go out there. They find any foreign molecule, they find any molecule, bring it to your lymphoid system and say, are you interested in this, okay? Now, if they find it in a hot spot, like in a cut or a wound, okay, they will bring information back and say, I found this in a wound, I think it's foreign, will you think about reacting, okay? If they find it kind of just lying around your body, they'll say, I found this, it was in the middle of you, it's probably yours, I recommend that you ignore it, okay? So dendritic cell will present a molecule, and it comes with a, I think you should fight this, or I think you should ignore this, okay? What has happened to people with MS is they've decided to fight their own myelin. So what if you then get a dendritic cell to give them their own myelin, but this time tell them, look, it's yours, okay? Ignore it, okay? So we can make dendritic cells tolerogenic. So what you can do is take cells out of people, you can make monocytes into dendritic cells with a combination of drugs. Interestingly, vitamin D and dexamethasone will make a dendritic cell tolerogenic. You then give it myelin, and then you then give these tolerogenic myelin-presenting dendritic cells back to the person. They show the person myelin, they say, this is yours, ignore it. And the T cells that want to react against the myelin fall asleep, okay, and become anergic. So you can use, so this is kind of reprogramming. This is exactly the opposite to what the cancer doctors are doing with dendritic cells, which is taking them out, showing them melanoma, okay, making them hit the melanoma, and then giving you these highly aggressive, so this is a dendritic cell vaccine as opposed to dendritic cell therapy. So who's doing it? Okay, this group are doing it, and they're giving you back your tolerized dendritic cells IV. In terms of how sexy this gets, okay, the Spanish are taking the same thing about, making you tolerogenic. They're then injecting them into your cervical lymph nodes. That sounds fun, okay? Because that's where the business end is, okay? That's where all the reactive T cells are coming. They're circulating in and out of your brain. So we're going straight into their programming area and putting the dendritic cells in there. Okay, in Antwerp, they're doing the same thing, tolerizing them. They're giving them into the angle of your neck so that they drain to your cervical lymph nodes. That sounds a little bit easier than injecting straight in, okay? And that's all out there. So that's kind of exciting, okay, and good to see. So if patients say nothing new is happening, there's kind of sexy stuff out there. Primary progressive disease, okay? Who knew? Yeah, ACTH, yeah, the 40,000 pound gel, okay? Makes creme de mer look like a bit of a gift, okay? You see, there's women in my family. I know these things. Who's impressed at my knowledge of, yeah. Sometimes I just go to watch the fish tank. Okay, so progressive forms of MS, same company, looking to see, giving it 3 days a month, okay, and then looking to see at month 36 what your walking is like. Okay, I just put this one in to remember that there are upper limb studies coming out there. We know that ocrelizumab hand is coming. It's not on clinical trials yet, but there is already a small study running, 35 patients, University of South Florida, investigator-led, looking to see whether we can slow upper limb deterioration in MS, now using this beautiful French upper limb outcome measure. Again, Jeremy was on this morning about repurposing cheap old molecules for secondary progressive disease. Okay, much as he is unique in his own right, this strategy is not unique. Calgary have been onto this as well, and they're looking at a couple of things, trying to repurpose hydroxychloroquine. Many of you know is in rheumatoid arthritis. Again, they have a neat study design. I say everyone's looking to get quick studies in this. Jeremy has this multiple arm design, which is good. They use this futility model, which is a fairly quick way of doing a small trial, and just really all this trial is saying, is this worth continuing? And the fairly strict criteria that at the end of a year, if it's not getting anywhere, you just stop. Okay, so it's a futility model rather than anything else. This, you may remember a while ago that there was a study about to be launched of dimethyl fumarate in primary progressive disease. That was the company-sponsored one that got pulled. However, the Danes are still running, I think, a little small study. Okay, I look at you because you're half Scandinavian, you might know. Okay. That's a bit like my granny's Irish, perhaps you know her. So it's, okay.
I can tell you the most wars we have had in Sweden has been with Denmark.
Okay, yeah, okay, we'll just let that one go then. Okay, so that's going. But interestingly, so their primary outcome in this one is not disability, it is neurofilaments. Okay, which gives me a time halfway through the talk to stop listing trials and talk a little bit about neurofilaments. Okay, I show a picture of Sharmili here, one because she's very nice, and two because I've stolen all of this from one of her presentations. Okay, I usually would put used by kind permission, but I don't think she knows. Okay. So, biomarkers in MS, there are a lot of them about there. I cannot deal with them all. I'm just going to talk about neurofilaments. So, neurofilaments, the nomenclature itself is tricky. They come in heavy, medium, and light. Okay, a bit like MS doctors. Okay. Neurofilaments medium are too unstable and you can forget about them, okay? Neurofilaments heavy chain, okay, are used in some studies and it's just worth checking through because if you read neurofilaments, you'll miss that sometimes it is on heavy. Neurofilaments heavy are released from damaged brain, but they hang around a long time. So they're a little bit like a sum of everything that's happened to you. So they correlate with EDSS. They correlate with disease duration. So the longer you've had MS, the more brain you've lost, the more NFH you have. So it's a little bit like your T2 lesion load, okay? It tells you how much has been damaged, and they hang around for a long time. It doesn't change all that fast, okay, but they are there. Neurofilament light, the excitement here is that this one is a rate of change molecule. It tells you how much brain is breaking down at the minute. Okay, so it'll come, it'll go again. If I was to hit you over the head, you'd have neurofilament light in your CSF for a while, and then it would go again. Okay, so it's a kind of a rate of change one, not how much change has already taken place. However, the good thing of that is that it is dynamic. The bad thing of that is it doesn't tell you what has damaged the brain. Okay, so if you have MS and you've had a relapse, your NFL goes up. If you have MS and you're losing brain through neurodegeneration, your NFL will be high. If you have MS and you fall and hit your head, your NFL will go up. All it tells you is that brain has been damaged recently. But clearly with the kind of drugs we have now that are stopping inflammation, it'll mean that much of that NFL that you're seeing is neurodegenerative. So the picture is clearing a little bit. Okay, the kind of information that came out in this, these are early studies, okay, that we'll see MS patients who've gadolinium enhancing relapsing remitting S has the highest levels of NFL compared to controls. These are just cross-sectional. You see these people are having a relapse in the first couple of days, not too many, okay? But as we start to get towards week 3, week 4, we can see that the damage that has been done, that dissolved brain is now flushing its way through the CSF. And in fact, you can measure that dissolved brain now in plasma as well, the bits that have leaked out. Okay, again, we'll see the more gadolinium enhancement lesions you have, the more NFL you have, and as you go into remission, the NFL falls. So this is quite exciting. It's a kind of measure, and I won't go into the full thing, just to say that the serum levels reflect the CSF levels, so we'll mix them up a little bit. We saw that with treatment, and this is why it becomes an interesting trial marker, is that if you put someone in natalizumab with relapsing remitting disease, okay, their NFL plummets essentially to that of healthy controls. Okay? If you get rid of the inflammation in secondary progressive patients, and this is how I interpret this one, it comes down, but there's still a little bit that you can't get rid of here, suggesting that you can get rid of the inflammation and that gets rid of some of the NFL loss, but the neurodegenerative element here, that slow background leak of NFL still goes on. Okay, taught us natalizumab, we'll see with fingolimod versus placebo a massive fall in the NFL levels down to almost what they would see as the upper limit of normal. Okay, and again, what we start to see, and this I'll come back to, is that the excitement with this in terms of a short-term trial marker is that the drugs we think of as better drugs seem to do better on NFL. Okay, and you'll see that the fingolimod NFL fall is much bigger than the interferon beta NFL fall, and these were matched patients in trials. So, again, just to kind of show that, can it be used to differentiate between drugs? Again, this is quite an old study now. This is all on fingolimod, okay? So if you were on no treatment, this is if you're on an interferon, okay? And this is if you're on fingolimod. Okay, so we can see the second lines go through. And this is the really interesting one. So this is patients with MS, okay, who, let me see if I can get this the right way around. This is their baseline, okay, and these patients were on interferons, okay. They then switched to fingolimod, and you can see that their NFL level falls, suggesting that fingolimod is doing better for NFL than interferons. These patients were on natalizumab, and then went on to fingolimod. Okay, and most of us will know that that's a move that you'll sometimes see activity, and you'll see the NFL goes up. So we've got this strong feeling that NFL might be able to sort our drugs out in terms of how well they work. And you can measure it quite quickly. These changes are quite soon. So what if you didn't have to do a 3-year head-to-head, but you could do a short study and just look at the NFL levels, okay? And this is, again, where Karolinska has helped us, okay? And this is non-peer reviewed. This is, and in fact, again, without permission, this is a photograph taken ad ectrems on my phone, okay? But what we have here, these are patients before treatment and after treatment, okay? And what you're seeing is dimethyl fumarate, fingolimod, all causing a fall in NFL. Look at what happens with alemtuzumab, okay? Massive fall, and with apologies to anyone who's a big fan, okay, if we look at the one first-line drug which has got here, teriflunomide, okay, suggesting that we might be able to sort drugs quite quickly by NFL. Okay, and again, I don't want to spend too much time on this. This is another big one. This is pulling all the Biogen study because they took levels in NF in all their studies. And what we find here is, again, placebo doesn't work all that well. Okay, Plegridy, we can see we're pulling a lot of people down onto the line, but look what happens with natalizumab. Okay, and these were at week 48. Okay, so again, within a year, but you can see that fall was probably sooner if they'd done it soon. So we might be able to use NFL very quickly to kind of sort out drugs where, and this is the one that really caught my eye, is that in the first year, if you have consistently low NFL, okay, you do not get new T2 lesions at the end of 4 years. So your NFL level over 12 months predicts where everyone's going to be at the end of 4 years, suggesting you could have dropped the last 3 years of this trial program, okay, if you just looked at the NFL at the end of year 1. And it's a simple blood test in this case, fewer lesions, fewer atrophy. The patients that stayed consistently over 16, okay, you can just see the difference in the new T2 lesions, and you can see the difference in the brain atrophy. So NFL is kind of coming through in relapsing remitting trials and in progressive trials as well. Slight difficulty here, you'll see the levels are generally higher, and they seem to be different in secondary and primary progressive trials. This is from Kapos. But again, you'll see about 12 months, we'll see the NFL result starting to show that these drugs might work, okay? This is EXPAND. Interestingly, it turned out a positive trial. This is INFORMS. It was a negative trial, okay? But maybe we had the wrong outcome measure because actually fingolimod was beautifully diminishing NFL and brain production and brain loss in this study, even though that didn't come through to a clinical effect. Okay, happy? End of teacher, right? Deep breath, here we go. How long have I got? Ooh, 5 minutes? Right. Right, we'll skip that one, that's dull. The next NIB, okay, another inhibitor. So I've told you that these are TNIBs, tyrosine kinase. This one targets one that is found mostly in mast cells, Okay, now mast cells are found in asthma and allergy. You would not expect them to have a big role in MS, but we've got this encouraging study looking at in primary progressive disease. Secondary progressive, okay, Jeremy's talked about, again, a little bit of repurposing, Calgary again, okay, the mirror Jeremy over there is looking at domperidone in secondary progressive disease. Neurovax, I've told you about this one already. This is the one against the peptides, okay, so this is the one that's looking to make you kill your own T cells which are reactive to myelin. It got orphan drug status in pediatrics and secondary progressive, so it's in trial here as well. Not going to surprise you that a large company that has a product coming for secondary progressive disease is doing a study showing that it is safe to chum from any DMT onto their product. Okay, suggesting that, again, if you're wondering where do you think siponimod is going to be placed, okay, I suspect it's going to be after another drug, and that the data will exist at that time showing you that it is safe to move from any drug you want onto siponimod. Right. Old dogs and new tricks, okay? Is there anything we don't know about Tisabri? Does it work at 6 weeks, okay, instead of 4 weeks? And that study is up and going, okay, to try and see whether we can make it safer by giving it less often. Okay, ocrelizumab, okay, and the infusions are there, but they're long. Can we give it over a shorter period of time? Okay, can you give ocrelizumab in people? Can we focus in on that? That's up and going, okay. Anything you haven't thought about doing with Copaxone? Okay, what about giving it once a month to people with progressive disease? Okay, and these studies are going up. It's not quite Copaxone, it's another glutamiracid from Mapi Pharma, okay, and they're looking to see whether that could come through. So again, even drugs that we thought had probably reached the end of their useful lifetimes are being looked to be repurposed. So, talked a lot about what we have. Are we certain we know how to use what we've already got? Okay, and a couple of interesting studies coming out, not looking at whether drugs work, but looking at how we use them, so starting strategies. Okay, and is it better to start low and escalate, or to start high and see how it goes? So the Liver MS study, this is for those of you that know Nikos, has piloted this along with Cleveland. Basically, a patient comes in, you say, I don't know whether you want to start high or low. You open the envelope and it says you are a high starter. Let's choose between these four. Okay, or you're a low starter, choose, and very pragmatically, just looking up to see what their brain atrophy is like in 3 years' time. Okay, again, too late to be unique. Okay, John Hopkins are doing very much the same again, randomizing people to early aggressive or first line and escalate. So these are messy real-world trials. Okay, there's going to be people swapping them. You can refuse after you've been randomized in the Nottingham one. So the data is going to be all over the place. Okay, but it might give us a little bit of a hint as to which of those strategies is best. Is it safe to switch between drugs? Okay, I don't want to limber in these. Can you go from natalizumab to teriflunomide? Okay, someone's looking to see whether that is a reasonable or safe thing to do. Might not be your first choice of drug to go to, but looking at it, okay, can you go from natalizumab to ocrelizumab? Okay, I suspect real world is going to tell us long before this trial ever reports. Okay, and can you go from natalizumab to alemtuzumab? We already know the Glasgow group have produced quite a nice real-world study in this, Answers MS, but again, this is being done in a rather more formal way, investigator-led, University of South Western with some response from Genzyme. So, starting and switching is fine. Stopping is really the kind of new frontier in MS. We can get people on the drugs. When do we take them off them? Interestingly, for the sake of time, I'm going to flick through this because Trevor and I have actually picked, out of Gordon's talk, we've picked this as one of the topics we want to deal with. Okay, but let's just say there are 2 studies going. One from the French called Stop SEP, Sclerose en Plaques. Okay, and then my favorite ever name of a trial, which is Discontinuing Drugs in MS, the Disco MS trial, okay, is coming out, and this is coming out from Colorado. Interesting, again, it would just pick up, so the French wouldn't even think about stopping someone until they were over 50, had 3 years with no relapses and no GAD, okay? The Americans don't think it's reasonable to even stop until someone is over 55, 5 years with no relapses and no GAD. NHS England, on no evidence whatsoever, said if you have an SPMS for a year, you should think about stopping, okay? Suggesting that money might be driving things. Okay, can I do remyelination in 4 minutes? Okay, so we've talked about stopping inflammation, we've talked about slowing MS. Can we make it better? Okay, So this really comes from, we talk about the molecules first, opicinalimab. So we know that people with MS do have the ability to remyelinate, but when the oligodendrocyte precursors try to get into the lesion, they're stopped getting in by a molecule called no-go, okay? No-go binds to LINGO on the cells. Anti-LINGO allows these cells to get in, and that's the basis of the Biogen study, encouraging first phase 2, big study up and going, add into your current therapy, outcome improvement, okay, not stopping it, okay? Interesting one, nanocrystalline gold tablets, okay, or cladribine you may think as well if you look at the cost of them, okay, but these are actual tablets of gold, okay, that you take to people who have a chronic optic neuropathy looking to see if we can improve their electrical outcome. And then another molecule, again, a monoclonal antibody, this time not going at the no-go LINGO system, but going at this RGN alpha inhibitor, again, trying to see if we can get precursor molecules in. Very encouraging animal studies, again, add on, looking to see, again, outcome is going to be improvement. So that's people who are already a bit knackered and you're trying to make them better. The neuroprotection model, however, is moving a little bit, that it might be a bit late at that point to do it. What we might need to do is get in very early after the damage and try to get myelin in at that point before any scarring has come through. So this is kind of what I'm calling neuroprotection. A couple of strategies. So clemastine, okay? Certainly all our patients know about clemastine because there was a phase 2 study showing that it showed remyelination in an optic nerve model. It is, however, one of the really old-fashioned sedative antihistamines, okay? But so long-term, if you remember, that group of antihistamines were replaced by the new ones because everyone fell asleep on them, okay? Not great for MS patients, but this is looking at maybe just to use it for a couple of months of treatment after an optic neuritis to see if we can get the myelin in, okay? Similarly, okay, this is a new beta-2 agonist. This is like salbutamol, okay? Again, being used. It's already used in horses, apparently. But it is now being used to see, again, after acute optic neuritis, to see if we can do it. And it's not going to surprise you. Who else is trying to get in this one? Yeah, the £1,000,000 gel, okay? Again, if you could rub some gel on, could we see some neuroprotection after optic neuritis? Stem cells, okay, I'm going to have to whiz over, okay, except to say that they are out there. Mostly what we're looking at now is mesenchymal stem cells. These are cells which are produced in your bone marrow. They usually produce fibrous tissue, cartilage, and muscle, okay, but these cells, the precursor ones, do a whole stack of stuff. They're anti-inflammatory, they're pro-remyelinating, they're pro-repair, okay, and we've got various grades doing. Bristol are just giving you a bone marrow extract into your blood, okay. This group, okay, from the Andalusia are taking the mesenchymal cells out and giving those back in, okay. Ottawa are taking them out, expanding them, and then giving them back in, okay. And this group from stem cell medicine are getting the same kind of cells, but this time from fat tissue, okay, and putting them back in, so you get a bit of lipo and your treatment all in one go. Okay, and this group are taking them out, differentiating them or redifferentiating them towards neural precursors and putting them back in. So if patient says, are there stem cell therapies going on out there? The answer is yes, there are, and plenty of them. Okay, so I'm going to whiz through these ones, okay? Bits on diet, I'll leave the slides up if you want them, okay? If you want to know the weirdest treatment someone could go for MS, okay, they can get their gut washed out and they can get someone else's stool from a non-profit stool bank. Okay, which I do like, and you can put in by colonoscopy, okay, and that's coming through, or you can get it once a month for 6 months, okay, that's coming through. And my final one, okay, the most left field one I could get is that there is a randomized controlled trial including nearly 500 patients giving them bee venom, okay, toxin weekly against them. So. I just showed you this. I was bored on the train. I did this on the train. Yeah, as I said to the guys, I think Pixar are safe when it comes to animation, but there is plenty out there going on. And 42 minutes, and we got through it. Thanks.